Okay, Dr. Babu, Department of Biomedical Engineering, Professor and Head, now Sri Sanma College of Engineering. Now I am going to explain about the internal electrodes. In previous slides, I will explain about the different types of electrodes. Surface electrode we have seen. Now in this class or in this slide, we are going to study about the internal electrode for measurement of biopotentials. Okay. Internal electrodes, it is used to detect the biopotential within our body. So biopotential or potentials are available within our body. The potential is to be measured or recorded to know about the pathology or normal of the internal organs. That's why we are going for the measurement of the biopotentials. So electrode used for the measurement of biopotential are different types. One is surface electrode, already we have seen. Now in this class we are going to see about the internal electrode and also micro electrode. So in internal electrode, one thing is percutaneous electrode, that is electrode inserted through the skin and going into the deeper seated tissues, that is the percutaneous electrode. Electrode itself or the lead wire crosses the skin. It crosses the skin and going below the skin. That's why this is called internal electrode. Also, it is also called deep-seated electrode. Entirely internal electrodes, connection is to an implanted electronic circuit such as a radio telemetry transmitter. No limitation due to electrolyte skin interface. Okay. So the surface electrode we are using the gel to reduce the resistance of the skin. In this case, there is no limitation due to the there is no limitation due to electrolyte skin interface. Electrode behaves in the way dictated entirely by the electrolyte interface. No electrolyte gel is required to maintain this interface because extracellular fluid is present here. So that's why in this internal electrode, as it is inserted into the skin, there is no gel is needed. The resistance problem of the skin is not needed here, not need to consider here. So that's why there is no gel needed here. Next, here we are seeing the diagrams of the insulated needle electrode. B is equal to coaxial needle electrode. Generally, in the internal electrode, one type is equal to needle electrode. In this needle electrode, again we are having the insulated needle electrode, coaxial needle electrode, multiple electrodes in a single needle, that is bipolar coaxial electrode, like that. So the first one is insulated needle electrode. Okay, so we are having the sharp edge that can be inserted in the skin. Next, this needle is insulated by a coating. This is the up and this connection is equal to wire, lead wire for connection. So this is the coaxial needle wire. Coaxial needle wire. Okay, next the C1, multiple electrodes in a single needle. Okay, many electrodes are available in a single needle. That is why it's called multiple electrodes in a single needle. Here we are having bipolar coaxial electrode. That is harmonic recordings using percutaneous wire electrodes. The D, the diagram D is equal to, there is a fine wire electrode connected to hypodermic needle before being inserted. Before being inserted into the skin, there is a fine wire is inserted in the hypodermic needle. Next one is E. This diagram explains about the cross-sectional view of the skin and the muscle showing fine wire electrode in place. Okay, the fine wire electrode will be placed into the muscle. So that is the diagram explains about the fine wire electrode is placed in place where we have to measure the wire potential. Then this diagram explains about the cross-sectional view of the skin and the muscle showing the coiled fine wire electrode in place. This is the, it's not coiled wire, it is a coiled fine wire electrode. Like this, we are having the internal electrode, we are having the different types of the electrode. It is only needle made up of the stainless steel and nickel. It is coated with the some insulating material. It is coaxial one. This is the fine wire. The fine wire is inserted before, the fine wire is already available before inserting into the skin. And this diagram explains about the cross-sectional view of the fine wire placed in the muscle. And it is the fine wire, that is a coiled wire. The diagram is nothing but a coiled wire acting as the electrode. Next, microelectrode. Generally, microelectrode is used for the measurement in the cellular level. Why we are using the microelectrode? Measure biopotential difference across cell membrane, inside of the cell and outside of the cell. Requirements: small enough to be placed into the cell. The electrode size should be very small. That should be placed into the cell. That's why it is called microelectrode. And also strong enough to penetrate the cell membrane. Okay, not only small, it has to penetrate the cell membrane. That's why it is having the strong enough to penetrate the cell membrane. Typical tip diameter is equal to 0.05 to 10 micrometers. So the types of the microelectrodes are solid metal, that is tungsten microelectrode, supported metal, metal contained within or outside glass needle, glass micropipette electrode with AgCl electrode metal. So these are the some examples of the microelectrode. So okay, this diagram explains about the microelectrode. Okay, this is connecting with the source for the lead wire. This is the tip of the microelectrode which can be inserted into the cell. So here we are having metal. The metal is insulated with the insulation. Okay, this is supporting shaft. Extracellular recording. Extracellular recording, that is typically in brain, where you are interested in recording the firing of neurons. So this is used for the EEG, that is firing of the neurons, that is spikes. Use metal electrode plus insulation goes to high impedance amplifier, negative capacitance amplifier. Okay, this diagram explains about the metal supported microelectrode. Metal supported microelectrode. So the first one is metal inside the glass. Outside we are having the Pyrex glass. Inside we are having the metal. The tip is sharpened. Okay. Inside we are having the metal, stainless steel, and outside we are having the glass, Pyrex glass. So this kind of microelectrode is called metal inside glass. Okay. Next one is the diagram B explains about the glass inside metal. Okay, inside we are having the glass. The glass is sharpened like this, and outside is covered with the metal insulation. So this metal film is available around the glass. After that we are having the insulation. This is the tip where the skin can be, the electrode can be inserted into the cell. So this is the types of microelectrode. One is metal inside glass, glass inside metal. These are also the metal electrode. Next one is glass micropipette. Micropipette electrode. How we are making the micropipette is we are taking the glass, that is okay, capillary tube made up of glass. After that, the center of the glass will be compressed like this by applying the heat. After that, this is cut into two pieces. Now we are having this kind of arrangement. On the one side we are connecting the metal electrode and it is connected with the lead wire for external circuit. On the another side we are having tip. This is filled with the electrolyte. So it is acting as the pipette. Okay. So if it is inserted into the cell and it is not direct contact with the metal electrode and the electrolyte transfer the biopotential to the metal electrode. Okay, this is the C. So intracellular recording, typical for recording from cells such as cardiac myocyte, need high impedance amplifier, need very high impedance amplifier, negative capacitance amplifier. These are some of the glass micropipette microelectrode. Next, motion artifact. We have to study about the motion artifact. Motion artifact is because of the movement of the patient. Sometimes the electrode will be moving, so that we are having the error in the measurement. So that's why it is called motion artifact. In addition to the off-cell potential, the electrolyte gel skin potential ESC can also cause motion artifact if it varies with the movement of the electrode. Because of the movement of the electrode, we are having the errors, that's why it is called motion artifact. Variations of this potential need to represent a major source of motion artifact in silver chloride skin electrode. This artifact can be significantly reduced when the stratum corneum is removed by the mechanical aberration with a fine abrasive paper. This method also helps to reduce the epidermal component of the skin impedance. However, the removal of the body's outer protective barrier makes that region of the skin more susceptible to irritation from the electrolyte gel because of the removal of the outer layer of the skin. Stretching the skin changes the skin potential by 5 to 10 millivolt. Stretching also, the skin resistance can be changed, that's why the skin potential will change to 5 to 10 millivolt, and this change appears as motion artifact. So this is measurement with the two electrodes. It is already we have studied about the electrode potential. This electrode, this is the equivalent circuit of the electrode, this is the equivalent circuit of the another electrode. It is both electrodes immersed in the body fluid or electrolyte. Now voltage developed in one electrode's electric potential. This is reference potential. The voltage difference between these two electrodes is called off-cell potential. This off-cell potential already we have explained. In that way, we can measure the internal potentials. Thank you.